Well, as the oldest person, probably the oldest person here, and having had this long, wonderful history with Richard as a postdoc in my lab, where I was able to, I don't know why he was the postdoc, but hopefully. I thought I mean, it's sort of, we learn more from our students than we doubtless learn from ourselves. And I had to think about what I was going to talk about. And should I write and make a pretentious old person talk? And I did. But I decided not to give it. And although I went and bought brand new socks and everything for this pretentious old person's talk, but I will give some advice, you know, as you expect from old people. And that is, eukaryotes are great to sleep with, but you don't want to do research on them. That was the only advice my daughter ever took from me. So it really is a truly great privilege being here and giving this talk. And I can't get the original title, but I did have an original and pretentious title. And I do think the original pretentious title is about great questions, and I did prepare that, and it lets me go on. I really am. I've been using computers since 1960. but I can't find the IBM cards, and that's the problem. I don't know why this thing isn't advancing. I think it's a good talk to give. And what I decided to do is talk about the latest research going on in our lab. And I think this is, maybe Richard would appreciate my doing this, in that does anybody know how to make this thing behave? It's not advancing. It's not. Oh, it's advancing on this.
Yeah.
If it's advancing for you guys, then it's advancing for me. All right. Great privilege, and it really is. It's too much fun. The whole thing. And these are the people in my lab. Most of what we do now is sort of has the potential of being useful, which and it's on antibiotics and antibiotic treatment and antibiotic. And these are the people in our lab involved in this whole, we call it the PKPD anti-mafia. The talk I want to give is about this, and this is really what's happening in our lab now. And Caroline convinced me this is the talk I should give. And I have all the pretentious things about this, and this is ribosome densities and bacterial growth, and trying to explain why some antibiotics are bacteriostatic and others are not, or bactericidal. A big, big question. And the other advice I can give you is it really pays to ask a big question and to be interested. It's far more important to be interesting than to be right. Nobody knows what right is, but we do know what interesting is. And I think this is a big and interesting question, and I'll try and convince you of that. And being wrong is not so bad. But I did present all these aphorisms and the kinds of things you would expect from an old person. And I will provide them with you. But the problem that I want to really talk about is this. That is to say, if we look at different antibiotics, and this really got us into an area that I didn't know. These are the bacteriostatic antibiotics, and this is Staphylococcus aureus. This is in the absence of drugs. This is treating with tetracycline, erythromycin, and linezolid. Very, very low rate of mortality, and so on. If we then treat with bactericidal antibiotics like gentamicin and so on, very high rate of mortality with respect to some. And so the question that got us into this is why clinically are these antibiotics of different types equally effective for treating why shouldn't the ones that don't kill be as effective as those that do? And I have a whole other talk about that. But the talk I want to continue on here is the question of how ribosomatic antibiotics do their thing. The molecular biologists give us these, you get the impression the answer is known. I mean, they have these wonderful diagrams, and then they have these very colorful pictures of antibiotics binding to ribosomes. I don't think the colors are right. I don't think they're that pastel, actually. But we get the impression that they really know, they really have their shit together, really know what it is. And then they'll tell you about binding and binding constants. But what you find out is they really don't know how antibiotics kill. And there's a great controversy going on with Jim Collins, because he suggested a general hypothesis, which was a very rude thing to do. He was outside the group. And so, he didn't do the experiments quite right, but at least he asked a general hypothesis. And I'm going to venture another general hypothesis about how ribosome binding antibiotics do their thing. It may be wrong, but I hope it's not uninteresting. And the worst thing about presenting a hypothesis is not that somebody rejects it, but rather everybody ignores it. And that's the worst possibility. The background behind this goes all the way back to the time I remember. I'm demonstrating that the relationship between the rate of growth is proportional to the number of ribosomes formed by a cell. Old work, I mean really old work, and at least Darwin didn't know these papers, but almost at that time. And Caroline really made it interesting why this is really interesting. I'm trying to convince people why there are great questions to be asked this way. And there was a recent paper by a group, I think the main author is this man Scott, who really did some really, really nice work addressing these questions. Now, this whole thing about the rate of growth and the number of ribosomes, there's a paper with, this is bacteria, this is allegedly what the ribosomes look like, I don't know. But anyways, there's some 50,000 ribosomes per cell. I mean, they're found in a cell. And the evidence is really quite good that the rate of growth is proportional to the number of ribosomes. That was the inspiration for the hypothesis. So the hypothesis makes the assumption that basically we have ribosomes which look like shrews. Maybe you would remember what shrews were in the cartoon from old days. And anyway, so antibiotics bind to them, take them out of the pool of effective ribosomes. And ribosomes are continually being replaced, the ribosomes binding are being lost. And at any given time, we have a distribution of ribosome numbers among members of the population. And as you have fewer, if you have fewer ribosomes, so this is say 50,000 here, if you have fewer, then you're growing at a lower rate. One very clear prediction, also based on really prediction. The assumption being that is if you have too few, you're going to be dying. And that the whole point that we're making is so that as ribosome numbers come up, we're down at this level, a fraction of the population is going to be dying. We now have a mathematical version of this model showing basically what it says. So that we're saying that as the number of ribosomes declines, we're getting into this position of having mortality. And so that some of the population is dividing, some of the population is being killed. The net effect is a low rate of decline. Now, how do you test that hypothesis? And which is, so that's a series of predictions. One is reducing the average number of ribosomes, which would be manifest as a lower rate of growth. We know that's the case. And the average number of ribosomes further reduced by the brine, antibiotics will become increasingly bactericidal. That is, bacteriostatic antibiotics binding to them should be increasingly bactericidal. And then because binding and associated to the extent to which they reduce the growth rate, it should be proportional to the concentration of the drug. So we make some very clear predictions. And also the corollary is that basically when you're coming out of lag, what you're doing, you have to do is produce enough ribosomes to start growing. So if you start with fewer ribosomes, it's going to take longer to get started growing, meaning if you reduce the number of ribosomes, you should increase the lag. And if antibiotics do the same, you should increase what is called the post-antibiotic effect. The term, that is, when you treat with antibiotics, it's well known that what will happen is you reduce the average, increase the time before it starts growing. So you wash out the antibiotics, we have this post-antibiotic effect. So the antibiotic, so the bacteria, even though there's no longer antibiotics, although there's an abundance of medium, it takes them longer to grow. This is a well-known phenomenon. We're suggesting if this hypothesis is correct, we should increase that. Now the question is, how do you reduce the number of ribosomes? Well, there was this wonderful collection of E. coli. E. coli has 7 ribosomal operons, RNA operons. And so that there's a collection where they deleted all but one of the ribosomal operons. You know, and if I only had one ribosomal operon, I'm sure I'd have a headache. But the question is, you can get these things to grow quite well, in spite of the fact that they have fewer ribosomal operons. Now, so that if we do a growth experiment looking at growth rate, you can see that the D5, the D6 is deleted for 6 out of the 7, D5 is deleted for 5 out of the 7, D4 is deleted for 4 out of the 7. You can see relatively little effect on growth. This is in a minimal medium with a bit of casein amino acids. The D6 doesn't grow well at minimal. D4 has a lower growth rate. D5 has even a lower growth rate, and D6 even a lower growth rate. So we can so it's consistent with that idea. And the lag increases as well. That is, as we reduce the number of ribosomes, the lag increases. So we have everything set, so the first assumption is correct. We then took the E. coli constructs. These are E. coli K12, I'm sorry to say, but that's okay. They're nice too. Not E. coli feeding. We treat them with tetracycline. D5 and D6 die off at a higher rate. Chloramphenicol, D5 and D6. The ones with only 1 and 2 ribosomal operons die off. Azithromycin and macrolide, same thing. So what we're saying is it's consistent with the hypothesis that if we reduce the number of ribosomes, well, we know only ribosomal operons, then we're able to make these drugs more bactericidal. The other thing we can show is that it becomes concentration dependent. That is, if I have 5 micrograms per mil, I get a less killing than I get at 10. And in all cases, the ones with the highest, with the ones with 5 and 6 are more sensitive to being killed in all cases. And the sensitivity, as you increase the concentration, you're getting greater sensitivity. Chloramphenicol is clearly the most prominent of this. Tetracycline, it seems to level off. And azithromycin, you can see 5 and 6 are being killed at a higher rate. Not a very high rate. Okay. And we can show that it is clearly not they're being more sensitive to bactericidal antibiotics because they have equal sensitivity to bactericidal antibiotics. So that it's not, and we should also point out the difference in rate. Let's say gentamicin is killing at a rate of 8 per hour. In the case of the others, it's 0.1 and 0.2 per hour. So it's very different. We also can show that basically we have this increase in post-antibiotic effect. That is, as you increase the number of deletions. So far, everything is copacetic. And the other line of evidence, I was having lunch with Christina Vege in Copenhagen, telling her about this. They work with Campylobacter jejuni, which only has 3 ribosomal operons. And as you can see, chloramphenicol and erythromycin are highly bactericidal. That's consistent with this whole idea. And then the question is, and with the molecular biologists, and certainly I didn't even know about ribosomes other than I told the students, and there's a comment by the book, I have to be molecular, who is not. Anyway, so now I am, sort of. I always was. Anyways, there's this, what is the relationship between number of ribosomal operons and number of ribosomes? And then this raises wonderful question, why 7? Why not 6? Why not 5? There's not much difference. And there's this quite wonderful study by a group of people who are not evolutionary biologists really addressing that question. Now I'm going to leave. You're welcome to the slides anyhow with all my aphorisms. Well, we asked the question by a thing called a bioanalyzer. My technician Ingrid McCall was terrific, and she isolated the RNA. I don't know how to do that kind of thing, and we sent it over to this machine that does bioanalyzing. And yes, indeed, there's just a relationship. As you increase the number of ribosomal operons, you have more 16 and 23S. So far we have a hypothesis we have not rejected. That doesn't make it correct, but it's certainly gratifying and probably publishable. Thank you.
How are the ribosomal operons regulated?
Well, that's a real wonderful story. I mean, there's obviously compensation. And these things are extraordinarily stable. I don't know. There's this whole regulation in response to growth conditions and media and things like that. It's an area that evolutionary biologists really should get into. I mean, it's an amazing thing. You go from the tuberculosis, mycobacterium has 1 ribosomal operon. And staph has about 6, E. coli has 7. Why? What is it all about? And it's so much related to growth and competition and everything. And I think it opens up a whole new area of delicious questions to address, I think.
It seems that the presence of antibiotics selection is quite strong to have more copies of these operons. Do you know what the disease effects are both in the presence of antibiotics and the absence of antibiotics, and whether that can also
Well, we haven't done that. I mean, what we've done is we've shown this relationship. We have to, the sensitivity of 16S and 23S isn't all that great. And are we actually measuring numbers of ribosomes? I mean, there's lots and lots of questions. If we keep them under selection with, say, tetracycline, will we get compensation? Because we know we're getting greater killing. So will we? This was my first attempt at this. This is not a question that we ask. We usually ask population questions. But I figure I'm now old enough to be a molecular biologist.